So today we are going to do photosynthesis in plants. So generally when we talk about photosynthesis, you have already learned it in the lower classes. So generally what you have is a rough idea in which carbon dioxide plus water in the presence of light is reduced to glucose and oxygen. So what happens is, now in the higher classes we know that carbon dioxide is reduced to carbohydrate, whereas water it is oxidized to oxygen. So generally photosynthesis is divided into two main steps: light reaction and dark reaction. But before we go into this, there are a few experiments which you have to know just to be sure what are the conditions that are necessary for photosynthesis. So the first one is to show light is necessary for photosynthesis. So before we start into this experiment, there are a few things you have to know. Generally when we do experiments on photosynthesis, you always have to destarch a plant. By destarch a plant, what it means is you have to keep the plant in dark for a period of 24 hours. So that what happens is photosynthesis did not occur in the previous day. So generally what happens is, we are going to take a well watered potted plant. We are going to cover the leaf, a portion of a leaf, with a black paper, and then we are going to leave it in sunlight for some time. Then after a few hours we are going to test this leaf for starch. Now what happens is, this covered portion does not test positive for starch. It does not test positive for starch, but this area it tests positive for starch. The simple reason being, these parts of the leaf were exposed to light. As a result, they perform photosynthesis, and the end product of photosynthesis is always starch. Whereas this covered portion, it was covered with a black piece of paper, so it did not show any positive test for starch. So this simple experiment proves us that light is necessary for photosynthesis. Another condition is to show that carbon dioxide is necessary for photosynthesis. So again, we are going to take a destarched plant. In this particular plant, what we are going to do is, half portion of a leaf is kept in a bottle or a flask containing potassium hydroxide. Because of potassium hydroxide, there is no carbon dioxide inside this bottle. Then we are going to keep this leaf in sunlight for a few hours. Then after some time, we are going to test this leaf for the presence of starch. Again, what happens is, this portion shows negative test for starch. This portion shows positive test for starch. Again, this portion did not receive any carbon dioxide because carbon dioxide was being absorbed by potassium hydroxide. But this portion, it was receiving carbon dioxide, it was able to perform photosynthesis, and hence it showed a positive test for starch. So from the equation it is pretty clear that we require light, carbon dioxide, water and chlorophyll. I have already done an experiment regarding light regarding carbon dioxide. The next is water. Water, it is very hard to show because if you do not supply water to the plant will eventually die. So there is no point in studying an experiment regarding the necessity of water for photosynthesis, but you can still do to show that water is necessary for photosynthesis by using radioactive water in which the oxygen evolved shows a radioactivity. But we are not going to into such details. So what we are going to do is we are going to show that chlorophyll is necessary for photosynthesis. So what we are going to do is we are going to take a leaf with patches of yellow and green. Such a leaf is called variegated leaf. The shaded portions of the leaf are yellow, whereas the non-shaded portion are green. This leaf, we'll keep it in sunlight for a few hours, then after some time we are going to test for starch. Now what happens is, this yellow portion shows negative starch test, whereas the green portion shows positive starch test. Due to lack of chlorophyll, what happened was this yellow portion of the leaf could not perform photosynthesis, hence it showed a negative starch test. But whereas the green portion, they contain chlorophyll, they show positive starch test. So we have already completed the experiments in which certain conditions have been proven to be essential for photosynthesis. Now the next thing we're going to do is we're going to go into the pigment systems, because like I earlier told you, photosynthesis occurs in chloroplast. A chloroplast is a double membrane structure. This is the outer membrane. This is the inner membrane. The inner membrane has certain extensions which form lamellar structure. This lamellar structure, when they are stacked together, they form grana, and grana is the site for light reaction. The inner portion of the inner membrane is also filled with a matrix. This is called stroma, and stroma is the site for dark reaction. This grana contains pigments. The pigments can either be chlorophyll or carotenoid pigments. The carotenoid pigments can either be red, brown. So what happens is the pigments, that is either chlorophyll or carotenoids, when acted upon light, converts the light energy into ATP. This ATP is utilized to further undergo the process of photosynthesis. So when we talk about pigments, light has various wavelengths. So photosynthesis always occurs best in wavelengths of light ranging between 650 to 680 nanometers. And this wavelength also represents red light. So this means that photosynthesis occurs best in red light. So what happens was, Emerson studied the various effects of different wavelengths of light on photosynthetic plants. Because of his studies, he gave you a simple Emerson enhancement effect. By immersion enhancement effect, what it means is, just remember earlier I told you that photosynthesis occurs best between 650 to 680 nanometers of light, that is red light. Now immersion, what it did was, light of wavelengths greater than 680 nanometer was superimposed on a plant also receiving light lesser than wavelengths of 650 nanometers. So because the same plant was exposed to light of wavelengths greater than 680 and lights lower than 650, the photosynthesis rate was greatly increased. If the plant was exposed to only greater than light 680 nanometers, the photosynthesis would not increase rapidly. Or if the plant was exposed to only lights of wavelengths lesser than 680, there wouldn't be any tremendous increase in photosynthesis. But if the same plant was exposed to the different types of light at the same time, that is greater than 680 or lesser than 650 nanometers, photosynthesis rate was greatly increased. Because of this increase in the photosynthesis rate, the exposure of the plant to lights of wavelengths greater than 680 nanometer and lights of wavelengths lesser than 650 nanometers is known as immersion enhancement effect.